Natalia Guerra, and I'm a PhD student in the Hospital Ramón y Cajal, Madrid, Spain. Today I'm going to present you my poster titled "The ICU Environment Contributes to the Persistence and Evolution of Klebsiella Species in the Hospital." As we all know, during outbreaks of pandemic clones, the surrounding of the patient gets contaminated. These contaminations are only studied during the outbreak. So, transmission pathways of SBL and carbapenemase producing enterobacteria are not well studied. This work investigates the dynamics of Klebsiella species within the environment of an ICU ward, which experience both clonal and polyclonal SBL carbapenemase outbreaks. So this work is part of the project Microflux, which aims to characterize the hospital microbiome and the dynamics of major opportunistic pathogens. We sample a large ICU of 14 rooms in 4 time periods, from 2019 to 2024. We sample in the first period before COVID quarantine, after COVID lockdown, and then in period C, we sample the ICU facility empty, because a new ICU was built and the patients were moved there. And then in period D, this ward was given another multifunctional use. We took 6 different samples. We took 3 dry samples from dry surface, which are the bed rails, the washing pot, and the touchscreen ventilator. Then we took 3 wet samples for where surface, sink drain, and sink pit trap. We then analyzed a collection of 1,487 Klebsiella, which we use antibiotic susceptibility testing, PCR, and clonal diversity testing. Then from there, we took a collection of 167 Klebsiella of different pulsotypes to sequence. We use different bioinformatician tools to do core genome analysis, resistome, and plasmid analysis. So, when we analyze the temporal and spatial distribution, we saw different species of Klebsiella. We had Klebsiella pneumoniae species complex, Klebsiella oxytocca species complex, and Klebsiella orogenes. We can see in these boxes all the 14 rooms along the 4 time periods, and we can see that Klebsiella pneumoniae in blue was the most isolated and more prevalent in this ICU. So we decided to analyze more into detail Klebsiella pneumoniae. As I can see here in this picture, it's the spatial, or again the 14 rooms, and temporal distribution of the clones of Klebsiella pneumoniae. We can see that a lot of these Klebsiella pulsotypes are isolated persistently. As we can see, this, for example, this blue one that is isolated from all the 14 rooms along the 4 sampling periods. So we had some ST prevalent, like ST307. Also in Klebsiella varicola, we had persistent clones like ST365-1LB. So when we decided to analyze more into detail the phylogenetic relationship, we saw in the phylogenetic tree that indeed we had the Klebsiella oxytocca species complex, Klebsiella aerugena, and Klebsiella pneumoniae species complex. We also analyzed the intrinsic and acquired resistant genes, here shown in this heat map, and we obtained some intrinsic genes like BLA-LEN in Klebsiella varicola, BLA-OXY in Klebsiella oxytocca species complex, and BLA-HAB. Then for the acquired resistant genes, we got BLA-BEL in Klebsiella varicola, BLA-BIN in many of these species, and BLA-OXA48, BLA-KPC, and BLA-CTXM in Klebsiella pneumoniae. Then many of these acquired resistant genes were in plasmids. So we decided to analyze the plasmids that we had. We identified a total of 925 plasmids, which we did a clusterization due to relatedness. So 639 were clustered in 60 plasmid groups that are shown in this picture. Only 286 plasmids were not clusterized and were in singletons that are not shown in this figure. So what we can see here is some clusters, plasmid clusters, were unique to each species, like Klebsiella pneumoniae, Klebsiella varicola, or in sometimes Klebsiella michigensis from the Klebsiella oxytocca species complex. Then what we can see is in yellow the clusters that have antimicrobial resistant genes, as I said before, like BLA-CTXM, BLA-KPC or BLA-OXA, and metal resistant genes like mercury, silver, copper, or arsenic. And then in green, we have the plasmids groups that only had metal resistant genes. We also had in some cases, in only one case, a plasmid group which had only antimicrobial resistant genes, and the rest of them did not have neither of them. We had some epidemic plasmids like the INC-F carrying BLA-CTXM with or without BLA-KPC, but we also had an INCL plasmid, INCL P-OXA48, like with the presence of BLA-BIM or BLA-OXA48 or BLA-OXA244, or lacking in any of these antimicrobial resistant genes. So in general, this plasmid group was also isolated from lots of enterobacteria in the sink. So in conclusion, the ICU patient care, which has a strong selection pressure, promotes sink contamination and long-term bacterial colonization. The coexistence of high number of Klebsiella population, both narrow and broad host range plasmids, persistent and availability of high-risk clones, and facility spread of resistant plasmids carry antimicrobial resistant genes against first-line beta-lactamase antibiotics. I hope you enjoyed my poster. Thank you for the founders and everyone that collaborated in this project.